So first, I'd like to thank the organizers for putting together a really great meeting and inviting me also to be part of this. And today I will show you some of our recent work on understanding tumor heterogeneity. And the first part of my talk will be on more like studying clinical samples, and then the second part is actually designing experimental models, trying to understand heterogeneity. And before I begin, I just want to acknowledge all the people who did the work. Vanessa and Michalina are two of the postdoctoral fellows who did all the clinical sample studies and developed the Starfish and iFish technologies. Andre and Doris has been working on xenograft models of heterogeneity, and we have a very close collaboration over the years with Franziska Mikor's lab and Mitat Gonen at Sloan Kettering. And these are the fellows in her lab, Philip Yukang and Lin Yu, who developed some of the mathematical modeling that I will discuss. So this just a brief introduction about what type of heterogeneity we discuss. So it's already mentioned that there is this inter-tumor heterogeneity that is between different patients. Then there is also heterogeneity between different lesions in the same patient. But today what most of us will discuss is this intratumor heterogeneity, which is heterogeneity within one tumor at any given time in the patient. And actually, if we compare normal and cancer cells and tissues, we could actually say that diversity or heterogeneity is the hallmark of cancer. And there are many different reasons underlying this, such as, cancers are genetically abnormal, they heterogeneous for genetic alterations. In normal tissues, the cells have a very different, very structured environment. Every cell in your body has a location. And in cancer, it's totally distorted. And also in normal tissues, each cell has a very defined epigenetic state that they cannot easily change because that would require a fairly large threshold. In contrast, in a cancer, these barriers are much lower, and as a consequence, in normal cells, there's a very robust architecture and very defined network of signaling pathways and expression patterns, whereas in cancer, it is very messy and very noisy. And basically, anything we can measure in cancer is likely to be heterogeneous. So genetic heterogeneity, immune response, epigenetic heterogeneity, and also the functional responses of cancer, they are likely to be heterogeneous. And I think one of the panel discussions we have is like, what is really the most relevant in terms of determining clinical outcomes? And I think a lot of it we still don't really know, but we can measure more and more. So if you actually consider incorporating this heterogeneity observed in cancers, like in contrast to this very traditional textbook view of how tumors evolve that seems to emphasize the clonal succession and these driver mutations that accumulate in cancer, but basically always focuses in on the dominant clone. In reality, we have a much more evolutionary biology view of cancer that seems to be closer to what we see in patients. We see a very extensive subclonal or clonal heterogeneity, and the selection and diversification in these clones is actually very important, and it's not so easy to study yet. So why do we even care about tumor heterogeneity? Mostly from a clinical perspective, it can be a prognostic or predictive biomarker for patient stratification. In general, there are many studies. I will show you some examples in our studies that higher heterogeneity seems to correlate with worse outcome. It can be also used as a tool to trace back tumor evolution, which in diagnostic samples, it's not easy to predict how the tumors evolved, but it's relevant for better understanding tumor evolution and also relevant for prevention, both primary prevention of cancer and also secondary prevention of progression to metastatic or invasive lesions. If we better understand heterogeneity, then ideally we can also develop better experimental models of cancer that really reflect the complexity of real patient samples as close as we can. And then lastly, it's also required for the individualized optimized therapy for everybody. And I will show you some examples in our studies that correspond to each of these statements. How can we measure heterogeneity? I mean, we will have a longer discussions on this, but very briefly, we can look at genetic, epigenetic, phenotypic heterogeneity, and the various combinations of these. And for each of these, there are global genomic scale technologies such as whole genome sequencing, whole exome, and also in situ or single cell technologies, both looking at single cells that are sorted or also looking at single cells in intact tissue. In my lab, we have been focusing a lot on developing technologies that allows us to assess genetic and phenotypic heterogeneity and also epigenetic in intact tissue samples in single cells. One reason for that is that I will show you some examples that we really think that the location of the cell and the changes in location and the interaction among neighbors, including the other cancer cells, and also the stroma microenvironment seems to be actually important. So we ideally would like to study tumors in intact environment. So we started these heterogeneity studies actually many years ago when this first, in breast cancer and other cancer types, this cancer stem cell idea was formulated. And in breast cancer, Mike Clark's group have published that there are these CD44 plus cells that seem to be more stem cell-like, and the CD24s were more luminal differentiated cells. So this study, which was published years ago, we purify these cells from normal and tumor tissues, and indeed they have very distinct gene expression profiles. But because cancer is a genetic disease, we also looked at genotypes. And then we surprisingly, should not be surprised, but we also found genetic divergence between these CD44, CD24 cells, such as, for example, 8q24 in this example was present in both fractions, the gain for 8q24, but then a gain for 1q21 was only present in a CD24 fraction, suggesting that it's not going to be like a simple, stem cell progeny model because there is also genetic diversity. So we extended these studies further, and again, we have used this immunofish technology, which allows us to look at copy number variations and also phenotypes. And again, this study, I just show you an example of, we looked at these CD24, CD44 cells and also copy number variations for multiple markers, comparing DCIS, which is ductal carcinoma in situ, is an early pre-invasive lesion, with invasive tumors. And I just show you here a nice example where we actually saw a clone that was a minority clone in a tissue in a DCIS CD44 cells that had a gain for this 11q13 region that became the dominant clone in invasive region, both in a CD24 and 44 fraction. So basically, this shows that there was a selection for these cells, and also there was a phenotypic switch changing the markers. And then, we wanted to look at more, quantitative measures of heterogeneity. And coincidentally, I went to the Galapagos in 2004, and I started reading this book, Beak of the Finch, which is an outstanding book. Everybody should read it. And then also this Measuring Biological Diversity, which is actually discussing how do you assess diversity in ecosystems. And in reality, a cancer patient is an ecosystem. So we started discussing things with Francisca, and then that's where we started these really collaborative studies on trying to apply these evolutionary and ecological models to understand cancer. And I went back to the Galapagos a few more times. So, for example, we want to characterize diversity. There is actually a very simple formula that ecologists use that characterizes measures diversity in a system, which is called the Shannon Index of Diversity. There's also a Simpson Index of Diversity. They're fairly similar. The formula is somewhat different, but it's a very simple calculation. Basically, you count how many different species you see and how many individuals in each species. And in a cancer, we can translate these species, a cancer cell with some unique identifiable properties that we can measure. So, for example, it can be a particular mutation, can be a copy number gain, and so on. And then we can apply the same formula for this. One of the key features for this is that you need single cell counts to be able to assess population heterogeneity in these tumors. So in this study that, again, we mentioned the DCIS invasive comparison, we applied this formula to calculate diversity of the different populations in DCIS and invasive. And then when we looked at this heterogeneity index, Shannon index, we saw that the basal-like breast tumors in general always have higher diversity, whereas HER2-positive and luminals have lower. And also the DCIS in invasive areas varied in terms of heterogeneity. Sometimes it increased, sometimes it decreased. Another kind of very useful plot to use that in ecology is this kernel histogram of species abundance, which again is a very easy way to visualize heterogeneity in a population. In this case, we apply this for this, looking at copy number variation in the different populations. And the x-axis is a copy number ratio for a particular chromosomal region. The y-axis is the number of cells, cancer cells with that copy number. And just by the picture, you can tell that this tumor is more homogeneous because all of the cell populations we assessed, invasive DCIS areas, have pretty much the same distribution of this population, whereas this tumor is much more heterogeneous because you see there is a very different cancer cells with different copy numbers. More recently, we applied this kind of studies, and this was published, so I just briefly go through it. It's trying to understand how heterogeneity influences treatment responses and also how the treatment itself changes the tumor in the patients who don't respond to treatment. In breast cancer, fortunately, we have neoadjuvant chemotherapy that's very commonly applied between diagnosis and the surgery of the tumor. So in this study, we studied the neoadjuvant chemotherapy patients. We had samples before treatment and also residual tumor in patients who did not have a complete pathologic response. And we applied this immunofish method that allows us to assess. In this case, we still use CD24/44. And then based on the presence of these markers, we differentiate the cells into 4 phenotypes based on, whether it's single positive for 44, that's the red cells, the pink cells have both, blue cells have only CD24, and then the white or gray cells don't have either markers. And then again, we looked at different chromosomal regions. 8q24 is the one we looked at most because that's very commonly gained irrespective of the subtype of the tumor. So it's useful to look at across tumor comparisons. And these are just an examples. We had about 60 patients, about 12 to 15 in each of the major subtypes, such as luminal, triple negative, and HER2-positive tumors. And in each one, we had a pre-post-treatment sample, except the 5 cases that had complete pathologic response. And then we basically counted at least 400 individual cancer cells from different regions of the tumor to assess phenotypic and genetic heterogeneity. This now we actually developed an automatic method to be able to do this, so we don't have to do it manually. When we just first looked at the Shannon index of diversity, combining all the tumors and all the cell types, interestingly, it was not significantly different between before treatment and after treatment. We saw some difference between a subtype, such as the HER2-positive tumors was a bit more diverse for this chromosomal region, and this was even more pronounced after treatment. But basically, the diversity index didn't change. It doesn't mean the tumors didn't change. It's just, they were diverse and they remain diverse. So it doesn't apply that there were no changes. And I just, we noticed that actually we do see significant changes in cell populations, like those 4 cell types that I told you about, the blue, red, and pink, gray cells. Interestingly, in most cases, we saw an increase in the CD24-positive cells, which are actually the luminal, more differentiated cells, and a decrease in the CD44-positive cells. And this just summarizes here. And of course, because this was a chemotherapy, this raises the obvious question is what is the proliferation rate of these different cell populations? And we looked at this by staining for KR67, which is a proliferation marker, in combination with a CD24/44, and this just shows some examples of this staining. And what we saw that in almost all tumors, in all cell types, proliferation decreased after treatment. So if we compare pre to post-treatment, there was generally a decrease. There were a few outliers where there was actually an increase, and interestingly, those were in the CD44-positive cells. And when we assess the fraction of proliferating cells in each of the 4 phenotypes, interestingly, the red or the CD44 plus CD24 cells were the most proliferative, regardless of subtype. So there is, of course, subtype-specific differences, such as triple-negative tumors in general are more proliferative. But even in this fraction, the 44 fraction was more proliferative than like the CD24. And even in the luminal tumors, we saw this difference. So that basically says that we see an enrichment for CD24 positive cells after in the residual tumors because those are less proliferative. The few outliers were where the tumor didn't respond, and actually because the CD44 plus cells were more proliferative, you can get the false impression that you're actually enriching for those, but in reality, you're not enriching. It's like they just don't respond. And lastly, I just show you like, why we like these in situ technologies. And we also developed algorithms that converts these confocal images to these computerized images of the tumor. Each cell is marked in a computer, and for each cell, we record the phenotype, like the 4 colors, and then also the genotype for like copy number gain or some other changes. And then in a computer, we can actually ask questions, is like, how this topology changes. And again, I don't go into the details, but you just show you some examples that, for example, this tumor, if we look at the genetic distribution of the cells in 3 different regions, before treatment and after treatment, just by this map, you can see that there are very significant changes, such as we see more of the cells with a higher copy number change, or also looking at phenotypic heterogeneity, we see more of the blue cells after treatment. And then we can also ask questions, is like, how are the neighboring cells to each other relatively genetically or phenotypically different, and also how the relationship to the stroma and how this may influence the responses. And lastly, just to go back and look at the clinical relevance of this heterogeneity, when we assess the pre-treatment heterogeneity in patients who had stable disease or partial response or complete response, the 5 cases that had a complete pathologic response had the lowest pre-treatment genetic heterogeneity, and this was significant even though the cohort was fairly small. But the stable disease and partial disease were no different and also didn't change during treatment. So just to summarize these findings, what we've seen is that intratumor heterogeneity varies according to tumor subtype, and this heterogeneity remains fairly constant during treatment, but this was a neoadjuvant therapy, and the pre-post treatment samples were basically the non-responders. So there was not really a selection bottleneck. The people who did not respond, we didn't have residual tumors. We saw that the proliferation rate obviously influences what cell types remain in the tumor. The slower growing cells were there. And then we also saw the topologic changes both for genotype and phenotype, and the more diverse tumors were less likely to respond to treatment. And another study we did, also was published earlier this year, I don't have time to show you, is we also compared primary and metastatic lesions, and in this case, the metastatic lesions, distant metastatic lesions were the more diverse and less different from one another than the primary and metastatic lesions. So now I'd like to show you a new technique that we developed, and it's really looks pretty, and it also gives me an opportunity to show you my starfish pictures. So we call it Starfish, and this is for specific to allele PCR combination with FISH. It's basically an in situ PCR combining it FISH that allows us to look at both copy number variations and point mutations. And the reason we wanted to develop this technique is because many of the cancer-driving mutations are point mutations, not just copy number changes. And again, ideally, we wanted to be able to look at these in situ in the cells in their intact environment. It's a fairly simple, I say fairly simple, but it took about 6 months to optimize each primers. Basically, we do a 2-step in situ PCR. In the first step, we have primers designed for wild type and mutant allele. I don't get into the details, but you do certain mismatches to make them specific. And then we also have an overhang that is not found in a human genome. It's a unique sequence. So then in the second round of PCR, we do the PCR with this unique overhang. And then the third round is actually, again, hybridization for the wild type or mutant probe with a color, like fluorescent color. And then at this stage, we also can incorporate FISH probes for other chromosomal regions. And that's how we get this Starfish method at the end. And then this allows us to look at multiple, both the mutations and also copy number variations. Current limitation is the limitation of the fluorotopes we can use, which is up to 5 to 7. But I'm hoping that Gary Nolan will present some of the in situ site-off that we could actually apply this to have a higher complexity of markers. This is just an example. So we developed this technique for combined detection of PI3 kinase mutation, the 1047, it's actually H2R mutation, which is the most common mutation in breast cancer, and combine it with HER2 because these two mutations frequently co-occur, and actually PI3 kinase mutation has been implicated in resistance in HER2-targeted therapies. So we thought that this is a good combination to detect. So we applied this technique to a cohort of 30 patients. Again, this was a neoadjuvant therapy. We had a pre-treatment diagnostic samples, biopsies. Then the patients received chemotherapy. It usually was a combination chemotherapy. A few cases also got Herceptin already in this stage or tamoxifen. Then we had the post-treatment samples. And then these patients were assigned to an adjuvant therapy, which was a HER2-targeted therapy, mostly with Herceptin. And then we had a follow-up like 3 to 8 years after this. So it's fairly, it's not a large cohort, but I show you some interesting findings that we had in this case. Just to show you an example, and I don't know how well this projects, but basically this is in one patient, the before-after PCR for a wild type. We mostly see the stroma showing up for the wild type primers. And then after treatment, actually we see more of the red cells, which are the mutant PI3 kinase, and then the FISH probe is the pink one, and then we have the merge signal here shows that we actually seem to have more of the mutant amplified cells after treatment in this case. So then we also designed an algorithm that automatically detects each of the cells. This shows here like each cell is circled by the computer, and then within each cell you count the dots. And then we get an automatic, count for, amplification, mutational amplification, mutation only, or wild type amplified wild type. So depending on what signals are detected. And then we get a list of, how many cells have what mutations in this. So these groups we call like a 5 species that we basically have in a tumor. And of course, in situ PCR has an error rate in terms of like probably not all the cells will work, as you see, because these are paraffin-embedded tissues, so there is some failure. But we assessed it that, because we're doing a sampling when we do the diversity calculations, so the sampling already incorporates that error rate that you don't necessarily assess 100% of each of the cells. So regardless of, some of the failures, as long as the failures are not biasing towards a particular population, it should be still accurate. Then we apply this to the cohort and then basically classify in each tumor. We also assessed multiple regions, 3 to 5 regions in each tumor to have an idea of the regional heterogeneity in the tumor also. And then we basically plotted this, what species we see before treatment. You see that before treatment, we don't see many mutant. We see a few mutant samples, but then post-treatment, we see many more mutant samples. And this is just kind of quantifying all the cohorts and the fraction of the cells. So we do see an enrichment for the cells that have PI3 kinase mutation alone or together with the HER2 amplicon. And basically, what was somewhat surprising, or maybe should not be surprising, but we actually saw cells with each individual change, meaning like just HER2 amplicon, just PI3 kinase mutation, and then also the double mutant cells as well. The wild-type cells most likely are stroma, because, the way we count the cells, we actually count everything. And then we see some of these other cell types with mutations. Then we again calculated the Shannon index of diversity 2 different ways. One of them is just looking at across species, meaning like, is there a change in these 5 species, the 5 cell types that we classify before and after treatment? And in most cases, there is actually an increase in the diversity, meaning like we're detecting more species after treatment than before treatment. And then we also looked at across area, which means that the distribution of these different cell populations within the same tumor in different regions of the same tumor, and this sometimes increased, sometimes decreased after treatment, and both were significant. Then when we looked at association with outcome of the patients in terms of recurrence-free survival, when we just looked at the species diversity, that actually was not significantly correlating with survival. The ones that had no change or had change were about doing the same. But when we looked at the diversity across area, then actually the patients who had significant increase or maybe even decrease, this decrease we only had 2 cases, so that may not be that significant, but the patients that had an increase in diversity distribution across area had a significantly worse outcome. So that means that in these patients, probably the different areas of the tumor selecting for cancer cells with different properties, and the more of these selections you have, the more likely that you have cancer cells that will eventually resist treatment. And of course, this disease-free survival is reflecting the success of adjuvant therapy, and we measured the primary tumor. But I think we will see to what degree the heterogeneous, probably heterogeneity of the dispersed metastatic cells corresponds to the primary tumors. But at least based on these studies, we see that based on the measures, diversity measures of the primary tumor, we have a prediction of how the patient survival will do. And again, like another utilization of these in situ technologies is to look at the order of genetic events. So in this particular case, we wanted to see what comes first, what is occurring first, HER2 amplification or PI3 kinase mutation. Of course, we cannot assess the other genetic changes which likely are occurring in the tumors, but we basically just wanted to see the relative order of these 2 events. And what we saw that the co-occurrence of these genetic events in the same cell are significantly higher than expected by chance, which would suggest that the cells with a double mutation have some fitness advantage, which was actually increased by treatment, meaning like the double mutant cells have even more fitness advantage due to the treatment. And then also considering the timing, the frequency of the cells we saw in the tumor, and also we had used some measures of proliferation rates of the different cell populations, we could predict that HER2 amplification is preceding the PI3 kinase mutations. And then we kind of just tried to come up with some hypothetical models that would explain why we still see all 3 cell types in the tumors. And one way, which we call the parallel model, it's like probably there is some initiating genetic event, which is not HER2 and PI3 kinase mutation, but something else, genetic or epigenetic. And then basically there is an equal probability to get HER2 mutation and PI3 kinase mutation, and then the HER2 amplified cells, acquire the mutation sequentially or vice versa, and then you still have C3 cell types. Or not, alternatively, we could have this HER2 amplification occurring as an initiating event, and then some cells acquire the mutation, PI3 kinase mutations, and occasionally it could happen that the HER2 amplification is lost. So there are examples that you can lose an amplification if there is no selection. Right now, we don't know the probability which one of these more likely to occur, but it's just basically suggests that you most likely can have all of the cell types in the tumor. Another nice feature of these in situ measures is basically we can determine the tumor topology. This is very similar to what you have on the mountain topology maps. These colors reflect the 5 different cell types, and then the circles and the closeness of the circles reflect the clustering of the cells. And what you see in this particular tumor, that there is actually an increasing clustering of the mutant cells after treatment, such as these particular areas seem to be closer to each other. And this can mean different things. It could mean that some population is proliferating faster or slower, so the cells would have tighter clusters, and also can reflect the migration invasion abilities of the cells. And it does look like that the mutant cells may have increased invasion and migration. But again, we can look at the spatial distribution of the cells before and after treatment. Interestingly, while these wild-type cells, which we think are stroma cells, basically we didn't see very significant clustering, and also it didn't change due to treatment. In contrast, the mutant cells had very high clustering, and it somewhat decreased after treatment, both the mutant and the mutant amplified cells. In contrast, the amplified or wild-type amplified cells, there was a lower clustering, but this increased after treatment. And again, this could mean that the mutant cells actually are more migratory and invasive. And Carlos Ortega created this transgenic model of HER2-positive PI3 kinase mutant breast cancer. And actually in mice, they have very nice observations that really parallel what we see in patients in terms of this heterogeneity and then the changes in heterogeneity in treatment. So now in the second part of my talk, which I'd like to also show you some examples of how we're trying to understand what does heterogeneity mean in terms of why do we see this heterogeneity in these clinical samples, and how can we understand the clonal dynamics that occur in tumors and explains why tumors maintain this heterogeneity and how we could use this to build better cancer models. So there are many questions that arise, from the studies that we do in human clinical samples. One is, why is heterogeneity maintained? If there is really a clone or subclone that is more fit than the others, why is it not just taking over the tumor? What is the reason to maintain minor clones in the tumor? What are the consequences of clonal heterogeneity? Meaning, like, are there some interactions among clones? And if there are interactions, are these direct interactions, meaning like the cancer cells interact with each other, or indirect, potentially mediated through the microenvironment? And then lastly, an important question in a clinical practice is how the treatment itself may change this clonal competition in tumors. So we decided to use a xenograft model of breast cancer, and the reason was that we wanted to understand the mechanistic basis of heterogeneity. So there are a lot of studies people doing now to do like single-cell barcoding from patient samples. Those are great. You will see heterogeneity. The problem is that it's very difficult to nail down the mechanism of the heterogeneity. So we decided to start from a breast cancer cell line, which is forming a very persistent but non-progressing tumor. Basically, like, the tumor remains about the same size up to like several months. This was in nude mice, in contrast to most of the cancer cell lines that form very fast progressing tumors. But this lack of progression didn't reflect lack of proliferation. These tumors are actually very proliferative. If we stain for Ki67, they virtually all positive, all the cells are positive. And for BRDU, which even for a few hour pulse, like 20–30% of the cells were positive. But this high proliferation is counterbalanced by high apoptosis and also necrotic cell death, as you see in this histology. So this, we thought, can give an ideal model to study clonal competition because we have high turnover. So for competition, you need replication. And at the same time, because these tumors don't grow on their own, anything that can make them grow, we will be able to detect, meaning like any clone or clonal combination, we will be able to detect. Then we designed clones that we generated from these cells. These are pools of cancer cells that we introduced a particular gene of that has been implicated in cancer. Initially, we started out with about 50 genes, including tumor suppressors, oncogenes. Then we came down at the end to 16 secreted proteins. And the rationale was that for secreted proteins, it's much easier to understand the interactions among cells. So we call these, the clones that have basically a pool of cells expressing a particular gene of interest. And then we did experiment when we mixed this clone against the parental cells. We call these the monoclonal tumors. And then we have other experiments when we mixed all of the clones or a subset of clones, and these are the polyclonal tumors. And then we had a PCR assay that we could detect the clonal frequency by using a clone-specific amplicon against a reference amplicon that is the same in all of the cells. Then we looked at these studies, and then we assessed tumor growth, metastatic ability, and also then analyzed the properties and clonality of the tumors. One was interesting first, and that if you look at these many green lines, most of the clones did not grow better than the parental cells. Pretty much almost all of them were about the same growth rate as the parental cells. Only 2 clones, CCL5 and IL-11, were able to drive the outgrowth of the tumor on their own, meaning like when we mixed 5% of these clones against 95% of the parental cells, these grew faster than the parental tumors. But then, interestingly, the polyclonal tumors, the purple ones, grew the fastest. So this included all of the clones mixed in equal ratios at the time of injection. We actually had to stop, sacrifice the mice at earlier time point because they reached the limit for tumor size. So that was kind of interesting, and it identified IL-11 and CCL5 as potential drivers. We looked at, like some obvious markers of proliferation, apoptosis, and vascularization among the different tumors to try to figure out why is this difference is observed. We did see higher proliferation of the CCL5, IL-11, and also the polyclonal tumors based on PRDU. Apoptosis was maybe slightly decreased, and also vascularization was slightly increased. I will come back to this when discussing IL-11 in further details. What was also surprising that the polyclonal tumors were not only faster growing, but they were very metastatic. Like even at 2 to 3 weeks after injection, when we had to stop the experiment, these were already spreading to distant organs like bone marrow, lymph node, peritoneal wall. We're continuing this study now, and we actually see metastatic lesions in the lung and liver developing very early on after the tumor evolves. The size of the tumor, obviously, you see much, much larger in a polyclonal, and also they're very irregularly shaped and very vascularized. These are the parental tumors. They're very small and hypervascularized. IL-11 tumors, they're big, but they're not as kind of vascularized and irregular as the polyclonal tumors. And then we found that FIGF, which is one of the factors, another factor that was in the mixtures, it was not able to drive the outgrowth of the clone on its own, but we think it contributes to the metastatic ability of the cells, and also these were much more vascularized. So when we mixed IL-11 and FIGF together, we actually can largely reproduce the phenotype of the polyclonal tumors. You see they're very vascularized and also growing faster, suggesting that there is some cooperation going on between these clones. In addition to seeing that IL-11 is able to drive the outgrowth of the tumors, we also wanted to know, is it required for the growth of the polyclonal tumors? Meaning, like, we knew it's sufficient, but is it required also? So we had these polyclonal tumors when we included all of the clones, and they grew very fast. When we removed IL-11, but had all the other 14 clones there, then these tumors didn't grow. So that suggests that even though the polyclonal tumors behave differently from the pure IL-11 tumors, IL-11 is still the main driver of this tumor growth. And again, I already mentioned that FIGF seems to be driving the metastatic dissemination basically by, looks like it just makes the whole tumor leaky. So basically all of the cancer cells can get out. Then coming back to clonality, we assessed the clonality of each of the tumor and plotted clonal frequency in the monoclonal tumors. These are the green ones. And then also the polyclonal tumors with all the clones or leaving IL-11 out. So these are the purple ones here. One of the interesting findings was that the clonal expansion doesn't equal with the ability to drive tumor outgrowth, meaning like IL-11, that was very potently driving outgrowth, it did not become the dominant clone. It remained the minor clone in the tumors, suggesting that, they are just the clonal dominance doesn't mean that that's the clone that's really driving the growth. Because LOXL3, for example, was the very good clonal competitor, but those tumors remained the same size as the parental tumors. And CCL5 was both a good clonal competitor and also driving the clone, and it likely because it enhances its own proliferation in the tumors. And then also, as you see, the green bars are much higher than the purple bars, and that means that each clone can compete against the parental cells much more efficiently than they can compete against the other clones. So even though these are essentially the same cell line, just expressing different factors, there is this polyclonal mixture that seems to limit clonal interference in a way that none of the clones can really dominate, but they seem to maintain some heterogeneity of clonality. Then we came back to the question, what would happen if we apply chemotherapy for these tumors to see how the clonal dynamics would change. So we used doxorubicin, which is one of the very commonly used chemotherapeutic agents in breast cancer, treated the polyclonal tumors with doxorubicin. So this was very effectively inhibiting tumor growth. We see the decrease in BRDU incorporation. And when we looked at the clonality, I just briefly summarize it. Basically, we saw that the good clonal competitors became even better, meaning like they could relatively expand more, and then the bad competitors became even worse. So basically suggesting that this chemotherapy would enhance the good competitors more. And we did see a decrease in Shannon index of diversity. That's because some clones became even more dominant in the tumors. So that's just something to think about when we trying to have treatment, and that treatment is not really curing the cancer. We may inadvertently actually accelerating the expansion of some of the cancer cells with the least desirable properties. Coming back to a question of how IL-11 drives tumor growth. So IL-11 has been a fairly understudied cytokine up to like 2 years ago, when there was like further papers suggesting that it's important for inflammation-associated colon cancer, prostate cancer, and some other cancer types as well. It has a very pleiotropic cytokine. It can act on cancer cells on its own, so it can have an autocrine growth factor properties. But at the same time, it also influences the immune system, multiple cell type, and also angiogenesis through influencing the endothelial cells. So obviously, it's not so easy to figure out exactly, nail down the mechanism why it's driving tumor growth. One experiment we did, which I don't show here, is we compared these polyclonal and IL-11 tumors in nude mice and NOG mice. NOG mice are more immune deficient, just to exclude the possibility that it's immune suppression is what we see. And we do see a somewhat faster growth even of the parental cells, but the difference between IL-11 and the parental is still there, meaning it's just not just immune suppression that IL-11 could be doing. I showed you earlier when we looked at the proliferation rates of these tumors, the parental and IL-11, the IL-11 expressing tumors were significantly more proliferative. It's a slight difference, but it's enough to explain the bigger size of these tumors. And this was actually independent of the frequency of the IL-11 clone in the tumors, because when we had like 100% IL-11 or 20% or 5%, essentially they proliferated, at the same rate, or the growth rate of the tumor was similar, again highlighting the importance that a tumor driver doesn't have to become a dominant clone, even if 5% of the cells can drive the tumor growth as much as 100%. You don't get actually more growth because of that. Then in order to address whether it's a cell autonomous or cell non-autonomous mechanism by which IL-11 acts, we did 2 different experiments. One of them is we downregulated the IL-11 receptor, the signaling receptor, IL-11 receptor alpha, in the cells that expressed IL-11. And in this case, compared to the control, like these cells, the ones that had the shRNA to the IL-11 receptor, they were still growing. I mean, the tumors were growing just fine, suggesting it's not an autocrine effect. And I'll show you later the computational modeling also confirmed this. And then another way we did this is we took breast cancer cells that had no measurable level of IL-11 receptor alpha. This is by RT-PCR. We could not detect the expression. But when we introduced IL-11 into those cells, it was still able to promote tumor outgrowth, suggesting that it's not a cell autonomous, but a cell non-autonomous effect, presumably through the microenvironment. And actually, when we looked at changes in the microenvironment in these tumors, for example, staining for collagen using this trichome staining, you see that the parental tumors are very different from the mixed IL-11 tumors. The matrix seems to be somewhat different. When we stain for SMA, for smooth muscle actin positive cells in a stroma, we see many more of these cells, but this occurs at later time points, so we don't know if it's cause or consequence of faster tumor growth. But what was really significant at early time points when we the size of the tumors were essentially the same, is the higher frequency of CD31 positive cells, which is basically angiogenesis, the blood vessels. This we saw a significant difference, even like 2 to 4 weeks when the tumor size is not significantly different, suggesting that IL-11 is enhancing outgrowth of the tumor by promoting angiogenesis. We don't know yet whether it's a direct effect or recruiting endothelial progenitors or recruiting some other stromal cells, which then recruit the blood vessels. We're currently investigating these possibilities. But this kind of raised the question is like, can we outcompete IL-11 in the tumors? Meaning like, what would happen if we mix IL-11, which is not a good clonal competitor, but it's a good driver of tumor growth, mixed with a clone that is a good clonal competitor, but not a good driver of outgrowth? I don't know if it's clear, but basically, LogCell3, I showed you previously, it's a very good clonal competitor, but the tumors are small, so meaning it's not able to drive the outgrowth of the tumors. IL-11, they are big tumors, and then we generated 2 different ratios of IL-11 plus LogCell3 clone, 1:1 and 1:18 ratios. And when we first looked at the growth rate of the tumors, just measuring volumes, in live mice, it actually looked like they are growing about the same rate, the IL-11 and the IL-11 LogCell3 mixtures, faster than the parental, but they're essentially the same. But when we collected the tumors, we actually found, and I don't know how well you see this, but this IL-11 LogCell3 1:18 ratio tumors, meaning like there was a higher fraction of LogCell3 than IL-11, they are actually look like a gigantic balloon. It's a cyst. And when we cut it up, and I'll show you in a picture in the next slide, it's basically necrotic tumor they lysed. Whereas when we mixed them 1:1, then it was still a solid mass, but not as fast growing as the IL-11 clones. And this just shows here that when we had IL-11 clones a different ratio, then the mass to volume ratio was different, and the clonality of the cells, obviously in the IL-11 clones, it was, the cells were there, whereas in a 1:1 ratio, we still had some IL-11 expressing cells when we collected the tumors. In the 1:18 tumors that seem to be necrotic tumors, it looks like that IL-11 was completely outcompeted by LogCell3, and that could explain of these kind of death of the cancer cells. So these are just the images of these necrotic cystic lesions that basically were filled with liquefied tumor cells and had very few viable tumor cells at the end, compared to the parental tumors that are basically large solid masses. So that actually shows that we can, a clonal competitor that's not able to drive the outgrowth of the tumor can outcompete a driver, and this can lead to the collapse of the tumor. To kind of depict it here, we can imagine that there is a clone that has a mutation, and this becomes a dominant clone, and it drives the growth of the tumor. But in terms of considering a heterogeneous tumor, this clone can be potentially outcompeted, and then that could lead to tumor collapse. Of course, this doesn't happen commonly in clinical practice, but it's just good to consider that could we modulate clonal dynamics in a way that we actually outcompete the worse clones and enhance the proliferation of the better clones that would be not so malignant in terms of their behaviors. So coming back to trying to explain, apply mathematical modeling for these tumors to explain why heterogeneity is maintained. First, Francisca group looked at what is the growth proliferation rate of these tumors, and it does look like that the tumors are exponentially expanding. And basically, when they looked at the clonalities of the cells and the proliferation rates of the cells in the monoclonal and polyclonal tumors, they could conclude that IL-11 expands both itself and the surrounding parental cells. So meaning it's not just expanding on its own. CCL5 is one of those clones that actually the tumor expands, but it's because of the CCL5 expressing clones are proliferating faster. It's not promoting the growth of the neighboring cells. Whereas IL-11 acts the other way. It actually promotes the growth of the other cells, not just the IL-11 expressing cells. And then they built models of considering how these two clones can interact. And very simplistic ways, each clone can proliferate or undergo apoptosis, and then there is a autocrine effect and a paracrine effect that can happen among the clones. And I don't get into all the details, but basically based on their prediction and based on the clonal frequency in polyclonal tumors, what was predicted that is the clonal interactions, meaning if your model incorporates interactions among clones, that can explain why heterogeneity is maintained. And also the diversity index with interactions remains high, meaning like the tumor remains heterogeneous. If there would not be interaction among clones, direct or indirect, so this doesn't necessarily say it's direct or indirect, then it was diversity would decrease in tumors. So basically, we think this is the reason why you have diversity in a way that the clones communicate with each other. So just to summarize this, what we see is that clonal heterogeneity is maintained in tumors. This is in the xenograft model I showed you previously. In clinical samples, we see this also. Interestingly, the clonal frequency doesn't reflect ability to drive tumor outgrowth, which actually makes it difficult to define what is a driver in a clinical sample, because we tend to focus on the most dominant allele. We're raising the possibility that maybe the minor clone can be a driver, and it's not so easy to determine what is the driver in clinical samples. It also looks like that if we have multiple clones, then they limit each other's ability to expand, meaning like a clone that would be, present if the other clones may expand less than when the others are there. This is relevant when treatment, when we consider in treatment, and sometimes treatment selects for a particular clone, that actually we can accelerate the expansion of that clone because the others are not there, even though the others, may be not responding to therapy. And then this, basically we eliminating this clonal interference, then we can accelerate progression. And I showed you also that the polyclonal tumors are more likely to metastatize, and then the driver of the metastatic spread and the outgrowth may not be the same, such as in our case, we know that IL-11 is driving the growth of the tumors, but FIGF is making them spread. So basically, you have at least 2 clones that may cooperate to drive this metastatic growth. And I didn't show you, but we have preliminary data seeing that basically the metastatic lesions are also heterogeneous, even though, it's basically most of them are not drivers and they're not metastatic clones. But if you have a subclone in the tumor that is able to make them spread, then basically it's like opening the floodgate. All of the cancer cells can go out, and then you can get heterogeneous tumors again recreated in the metastatic lesions. And then this interaction among clones is what maintains heterogeneity in tumors. And of course, this was a xenograft model. What we would like to do now, and I think this is the challenge for every one of us, is how we can show this in clinical samples, and more importantly, how we can use this knowledge to better design treatments for patients that would consider these dynamics of the tumors. And with that, I'd just like to acknowledge all the people who did the work I already mentioned: Andre, Vanessa, Michalina, Doris, and then also Hee-Jung Kim and Soyoung Park initially developed the immunofish. We had a fun and very productive collaboration with Francisca's lab, Yu Kang, Philip, and Lin Lu over the years in her lab. And then we also have many, many outstanding and clinical collaborators virtually around the world. Pedra Margani and Sarah at Johns Hopkins, with Norway, Annelies Boresendale and her group, Hege, and then also with George Rice-Philo, who's now at Sloan Kettering and also from Barcelona. And then Shalev Itzkovitz with MIT actually helped with developing some of those algorithms for the computerized images in the what I showed you in topology. And with that, thank you for your attention, and if there are any questions, happy to answer.
So to get the ball rolling, Dan Kapp, excellent presentation. In your spatial studies showing spatial differences pre and post neoadjuvant treatment, were you able to correlate that with blood vessel location? Were they closer to the blood vessel, or were changes seen at the periphery of the tumor versus central tumor?
We haven't done that, but that's one of the directions we're going to look at the microenvironment and how that could, influence this clustering of the clones, and not just the blood vessels, but we're very interested in leukocytes, because particularly for HER2-targeted therapy, there is a large, CD8 cell response in these tumors. So we're doing some of those studies now. And
the correlation with the immune response around the tumor with outcome.
Yes, so that's one of the directions we're continuing the work, yes.
So I'd like to ask, in setting up your battle of the clones, you made choices. You didn't pick randomly from all the possible genes and products that could be made by tumors. So could you talk a little bit about why you chose those 16 secreted molecules? Oh, so,
we came down to initially we just assembled a list of about 50 genes that have been implicated in cancer based on either they mutated or overexpressed and have been associated with some metastatic progression or angiogenesis. So the 16, actually it was 18, few of them dropped out, like IL-6 dropped out because it had a very strong systemic cachexia effect. But we basically picked based on published profiling of tumors, that they either overexpressed in particular breast cancer or have been implicated in metastasis or angiogenesis. So that's how we kind of had. And you can look at the full list. It includes many VEGF. Interestingly, VEGF ABC was not driving the clones, and even VEGF-D is not making the tumors grow, even though they're angiogenic, but they would not grow. That's kind of interesting that, recruiting blood vessels may not be enough to grow, at least in this kind of model, yeah.
So in a sense, it was interesting for the ones you ruled out as well as the ones you ruled in. So for the ones you ruled in, let's say IL-11, can you tell us more about IL-11 and human breast cancer? Real specimens from patients? So
in patients, it has been very poorly studied, partly because availability of reagents. So we ourselves tested the antibodies against IL-11 or IL-11 receptor. There are some published studies. In our hands, those antibodies don't really work, meaning like if we knock down IL-11, we still see the signal. So I usually don't believe those antibodies. So I didn't think it has been evaluated. For expression profiling, coincidentally, it was left out of the adjuvant arrays. That's also kind of funny because it's not funny, but we wanted to look at this Metapric data set, and it's not there. I mean, how could you leave out the gene? But it was left out. So then we looked at some of the other expression data, but it's really difficult to see correlations because, when if you're looking at array data, if you have a minor clone, it may not show up as overexpressed. And then you don't see the associations. This actually made us think about all these associations we do with expression. You probably looking at the dominant clone or the most prominent cancer cells for expression. And these minor things, the same for FIGF. It's almost no studies. I mean, it's shown to promote metastasis. But in terms of correlations with outcome, it's difficult to study. And again, it can function as 5% of the cancer cells can already promote metastatic growth. So you would not see it maybe if you just grind up the whole tumor.
Yes.
Oh, Alex Hughes from UCSF. I really liked the talk. And I think following on from this question, you mentioned briefly that you were initially interested in some of the canonical breast cancer mutations, but they didn't really follow through into the later work. And I'm interested in, is that because kind of the phenotypes were difficult to put quantitative values on, or because they didn't have any particular effects that were interesting?
So the one that was able to drive growth, which I remember is KRAS and I think it was HER2. But first of all, they were growing too fast, and also it looked like all cell autonomous. So we thought that's not as interesting and would not allow us to really study clonal dynamics because it would just, grow up too fast. But I think it's interesting to think about whether those, what we consider cell autonomous mutations, do they have paracrine effects? And most likely they do, because many of those, for a tumor to grow, you need the blood vessels, you need the microenvironment. So they must have some cell non-autonomous effect, either directly or indirectly. And I think it will be interesting to study those. If you put in a mutant gene, is that only cell autonomous, or you can have some paracrine effects?
Thanks.
Yes.
Yes.
Nellie,
Yes.
it was a great talk as ever.
Thanks.
So I'm very clinically driven by this. Your neoadjuvant observations.
Yes.
So the expansion of that smaller HER2 amplified PIK3CA mutated population during neoadjuvant therapy, that makes complete sense.
Yes.
The CD44 one I'm more confused by. So on the first slide you showed expansion of CD24, the luminal, and a decrease in the CD44.
Yes.
I thought it was.
Yes.
If you look at Jenny Chang's stuff, which was published a couple of years ago,
Yes.
she showed, I thought she showed an expansion of CD44. I don't know. She commented this in terms of EMT. What do we take out of this? Is this dominantly in the triple negatives, as she reported? What do we take out of this in terms of neoadjuvant therapy? How can we monitor this for development of resistant subclones?
Okay, so there were two major differences between her study and what we did. So if you remember, she did FACS.
Yep.
And did not actually confirm what is a malignant cell and not.
Yeah.
So knowing CD44, that it's a very ubiquitously expressed, it's really important to confirm. And I know they did gating for the leukocytes and so on, but it's very hard to gate out all the stroma. So we did everything in situ. We actually saw the cells. Also, we only counted the cells with a genetic alteration, so we were sure that those are cancer cells. And then if you see in this, so in terms of the proliferation, it actually makes perfect sense. You know that ER-negative tumors are always usually more proliferative than the more differentiated luminal tumors.
And more responsive.
And more responsive to chemotherapy. So what we see actually fits with what we see in a clinic in terms of like the less differentiated cells seem to be more proliferative. There was another study published by a Korean pathologist, So Young actually, who's seen that residual tumors in non-responders can have more CD44 cells. But then if you looked at the ones who had pathologic complete response, they had higher CD44 ratio. So that's why I was trying to show this paradox here, that we had non-responders, and interestingly, in those, the CD44 fraction was more proliferative. So you could see some paradoxical enrichment for those cells, but that's just because they don't respond, and then they proliferate more. But in the other ones, like in particular in the luminal tumors, we do see an enrichment of more CD44 cells, and that was more pronounced in the luminals.
So your last line there, that fast-growing resistant population, what exactly is that? That's CD44 positive, or no?
Which one?
Your fast proliferating resistant cells, chemotherapy enriches for.
Overall,
what are the characteristics of that one?
Yeah, so overall we do see more CD24s, but in these few cases that I highlighted here, you can actually see CD44 cells in the tumors. So I think what we like to highlight here is that you really have to look at the proliferative fraction to make conclusions, because obviously whether the cell responds or not to treatment and how the proliferation varies according to phenotype. And you can use any marker that you want here. It's not necessary you have to use CD24/44, but that's the way to assess what is responding, what is not responding. And in a way, we know that the differentiated cells tend to not respond.
Yep.
So it
Great.
kind of fits. Yeah, thank you.
Yep.
Yes, Mina.
Nellie,
Yes.
beautiful talk, beautiful
Thank you.
talk. The question I have for you is, what's the role of normal cells in tumor growth? Because as you know, there are a lot of people, some pathologists for years and years and years, who say without the normal host, normal cells, the tumor will not grow.
Yes.
So when you're doing this, how could you distinguish the dormant cells, the tumor dormant cells from the normal? And while you're looking at it, what % of the tumors actually have normal cells within them? Let's just start with saying non-growing cells. Then you can answer the question about dormancy versus quiescence.
So in this case, like the clinical sample studies, we do see stroma. And, like when we did this Starfish study, for example, this included counting all the cells. So that's why I was emphasizing that these wild-type cells probably reflect the stroma. The ratio can change. And then the second part of my talk, that was exactly the point I made, that it's actually the microenvironment what makes the tumor grow. And it's basically the cancer-driving clone recruits the microenvironment, and that's how you have a cell non-autonomous effect. So the normal cells are really important for the tumors to grow. And then, in this particular case, we didn't look at in detail in terms of like the stromal interaction, how the influence changes, but I'm sure it influences, and that's what I said, that's the direction we're going now, to try to understand how the stroma cells interact and influence the treatment and responses.
But as you well know, when you put the tumor cell and a xenograft, even if you add the human stroma, they disappear.
Yes.
And what comes in is the mouse stroma.
Yes, yeah,
right?
we see that.
So, yeah. So is that what you're counting as wild type?
So in the xenograft study, that was not, we didn't apply these mutation studies there. Here, when we look at the clones only amplify the cancer cells. So in this case, in this staining, we did count the stroma. I mean, we looked at the stroma and the CD31 also. We look at the CD131 positive cells, but like we had the clinical studies where we looked at amplification mutation. In that, we did count the stroma. That was presumably the wild-type cells. And then here, we looked at the stromal cells. So now we're trying to integrate everything, to have stromal markers combined with cancer cell markers. We're kind of running out of the colors, so that's why I'm really hoping that the site of technology, I really want to, there was a paper published by a Swiss group that you can apply the site of kind of method for in situ, so you could go up to 30 markers.
Well, wait for Gary Nolan. He's going to give you 70 of them.
So we're very interested in trying that out, because it would be really nice to do this in situ things now with like 30 markers, including combination of mutations and stroma. Yes.
Yes, Paul.
Yes. Yeah.
Beautiful talk. I just wondered if you could comment on the relative effects of chemotherapy versus other therapies, because of course when you say that you're selecting for a slower growing cell, if you're using chemotherapy, there's an obvious mechanism there, right? I mean, the cells perhaps that are growing faster are going to be more susceptible to chemotherapies because they damage DNA. So there's also some fairly simplistic sort of things that are going on there, rather than selecting for a more or less differentiated cell, right?
Yes.
You're actually selecting a cell that's resistant to that particular therapy, right?
Yeah. So in this study, like I didn't want to point it out, but in this Starfish study, we had 2 patients who got, 2 or 3, who got Herceptin in addition to the chemotherapy, even in the neoadjuvant setting, and we had 3 who got tamoxifen. And in those cases, the ones who got Herceptin and did not respond, there was a very clear selection for a resistant population. So there was a decrease in diversity. So that would suggest, and this has been observed multiple times, that targeted therapy actually accelerates for selecting for a clone that's resistant. We didn't have enough patients to conclude that, because it was only a few samples. But
you're talking about the patients that received HER2 therapy alone, or received the combination?
Together with chemo.
Together with chemo.
Together with chemo. So what we're doing now, it's very hard to find those cohorts, but we're trying to compare the different types of HER2 therapies, and also chemotherapy is still commonly used as a neoadjuvant, even in HER2-positive patients, although nowadays they don't apply it alone. In most places, you combine it with Herceptin or HER2-targeted therapy. And based on our studies, it makes perfect sense. If you're doing chemo alone, you may actually select for the cells that will be resistant for HER2-targeted therapy, because you're enriching for the mutant cells. The PI3 kinase mutant cells seems to be less proliferative. And also we see that some of those don't have the amplicon, so they're not going to respond. So that, again, just shows that it's really important. It would be nice to look at these cell populations before you start treatment, and especially you don't want to do a treatment that could accelerate selection for resistance. But that's one direction we're trying to do now, is like compare the different types of HER2 therapies. Very hard to get a cohort where you have HER2 targeted therapy alone without chemo. It's usually combined. But I think we have a few cases where we could look at that.
Right. It also, actually, the idea that combining therapies will be more effective is an obvious thing. And in the melanoma trials, where people are now combining BRAF inhibitors with MEK inhibitors and other inhibitors of that pathway, right? You're going to theoretically be able to, prevent outgrowth of those clones that have resistance, right?
So the other one, what we really like to be doing with Francisco is to try to not only design combinations, but define the best way to combine two drugs, meaning like considering the responses and maybe do a different sequence, because even that could make a difference. Like in this case, probably you would want to do HER2 first, HER2-targeted therapy, then add some chemo and maybe vary it in different ways. So I think that's very interesting and should be explored more, because I think a lot of the currently used drugs could be more effective just by administering them better. And Francisco had a very nice paper on even radiation in gliomas, that just changing how you spread the same radiation can make a difference in outcomes. So, yes.
Nellie,
Yes.
Excellent talk. So you outlined nicely how your battle of the clones can give you an idea of the sense of the role of normal cells and the sense of phenotypic heterogeneity. But you also said that some of the most potent mutant drivers were too powerful. So I guess the question is whether you feel that that might account for that. If you look at the list of mutated driver genes, they tend to be overwhelmingly cell autonomous, at least at their core function. Obviously, you have to have, as you outlined nicely, you have to have some effects on, because you can't grow without normal stroma and so it seems to be cell autonomous. So do you think you would get a different answer? The clonal competition would be different if you used these cell autonomous classic mutant driver genes?
Maybe you do, but like one of the problems also that we're defining drivers based on mutations. And maybe even in those tumors, you can maybe upregulate a chemokine or cytokine. So, what I'm trying to say that, yes, we see that those cells are mutant, and you see a dominant allele, but that could be due to because you could secrete something that then have a cell non-autonomous effect also. I think that would be something really interesting to address. Ideally, because you want to minimize the genetic heterogeneity, so it would be really nice to use like a CRISPR technology and start out with a cell and put in defined alterations. Of course, it's not really the patient sample, but it would allow us to look at some of these questions, address it. And yes, I mean, probably many of the mutations have cell autonomous autocrine effects, but there could be also non-autonomous effects. And even in this case, I didn't get into details, but the double mutant PI3 kinase HER2 mutant cells behave differently from HER2 amplified cells only. They actually tend to be, and that's in Carlos's study also, they tend to be more invasive, more metastatic. So then, the metastatic lesions probably even more enriched for them than what we see in a primary tumor. Again, it would be better to study a lot of those. Yes?
It really was a super talk. So how are we going to go about identifying these subclonal drivers that are working in a non-cell autonomous fashion that, are going to evade detection from a, perhaps a single biopsy or be subclonal below the detection of a conventional sequencing approach?
It's really a challenge. So some of these technologies that allow you to look at single cells, ideally, in situ, would be really nice to apply, but of course you have to know what it is. So we're thinking about different ways to go about this. One is that you could do a bulk profiling or regional profiling, really high depth to find these, and then go back. And then in order to determine what is the driver, you really have to follow clonal dynamics. This is what people in classic bacterial yeast genetics did, is like follow clonal dynamics over a long time. And in patients, ideally, would be nice to do this by repeated sampling. It's difficult to do from the tumors, plasma sequencing or CTCs, if they reflect what's going on in a tumor, could be one way to do that. And then you would see what's changing. And then at the end, we have to understand the function. So what we're proposing, and of course it's not ideal, but you could take the tumor of the patient, derive xenograft from those in a way that you maintain heterogeneity, and we doing it in a way that we basically, it's a complicated story, but basically we want to put in a known driver. So basically we don't select for xenografts because, one of the biggest issue with preserving patient samples that you're selecting for cells that grow in a mouse. Yeah. But if you remove that selection step, then we can hopefully maintain heterogeneity even more. And then we can couple with barcoding and then look at minor clones and the behavior. Of course, it's in a mouse. It's not perfect, lacking the immune system. But, I think really we need a combination of some, following the patients as much as we can through treatment and combined with some functional understanding. But that's what I was saying. This is the challenge for all of us, how to figure this out in patients.
And just very quickly, how well do you think the GEMS models, let's say BRCA mutant breast cancer, represent the heterogeneity you see in BRCA mutant triple negative breast cancer?
So that has been one of the problems with the GEM models, that heterogeneity is usually not there. Because they don't have genomic, BRCA1 actually may have some genomic instability, but you have multiple problems. One of them is that you're putting in a very dominant gene or knocking out the gene in many cells. It's not like a single cell. Some of Tyler Jax's models is better, that at least you start from a single cell. And then probably would want to put in some, something that increases genomic instability, like rondopinol seen in the telomerase knockout, that you get more heterogeneity. Because traditionally they haven't been very heterogeneous, the mouse, most of the tumor models. And that could be one of the reasons why they haven't been so predictive. Yeah. Okay. Good. Thank you.